All right, let's jump right in with a classic exam question, one that pops up all the time. If a DNA sample has 30% adenine, what's the percentage of cytosine? If that makes you pause even for a second, don't you worry. Just stick with me through this explainer because by the end, you are going to absolutely master the structure and packaging of DNA. This is a guaranteed high-scoring topic, so let's go get those marks. So, why is this topic so incredibly critical for your exam? Well, because this isn't just some fascinating biology puzzle. It is a high-priority, 100% scoring zone in CUET. You can bet you'll see direct questions on things like Chargaff's rule, maybe some diagram-based questions on the double helix, and for sure some conceptual traps around this very problem of DNA packaging. And this number right here, 2 meters, that is the insane length of DNA crammed inside just one of your microscopic cells. Seriously, think about that. Understanding how this incredible feat is even possible is your direct ticket to locking in at least 2 or 3 questions. This topic shows up almost every single year. Getting this right is completely non-negotiable. Okay, let's dive into this thing. We are going to unravel this 2-meter mystery piece by piece. We'll start at the very beginning, at the molecular level with the structure of the DNA thread itself, and then we're going to build our way up all the way to that fully packed chromosome that you always see in the textbooks. You see, to solve the packing problem, we first have to understand the material we're working with, the DNA itself. So we're going to cover its fundamental structure, the super important rules of how it all connects, the different bonds that hold it together, and then, finally, how it's all packaged up. Let's start with the absolute foundation, the double helix model. This was famously figured out by Watson and Crick way back in 1953. This is the fundamental architecture of our genetic material, and you better believe every single feature is a potential exam question. Now let's break down the key features you absolutely need to know. First off, the 2 strands are anti-parallel. What does that mean? It means they run in opposite directions, kind of like the 2 lanes of a highway. The outer rails of this twisted ladder, that's the sugar phosphate backbone. Tucked safely inside, forming the rungs of the ladder, are the nitrogenous bases. And remember this little detail? It's a right-handed coil with exactly 10 base pairs for every 3.4 nanometer turn. So what are those bases on the inside anyway? Well, they fall into 2 families, and this is a classic point of confusion for students, so listen up. We've got the purines, that's adenine and guanine, and we have the pyrimidines, that's cytosine and thymine. Knowing which base belongs to which family is tested all the time, so pay close attention right here. So how do these bases actually connect? Is it just random? No way. It's a perfect, incredibly precise partnership, and it's governed by a fundamental rule of biology that you absolutely positively have to know for your exam. And this brings us to the great Erwin Chargaff in his groundbreaking discovery, which we now call Chargaff's rule. What he found was that in any DNA sample, the amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. This is literally the bedrock of how DNA works. So the crucial point is this simple formula. A always equals T, and G always equals C. And this directly answers that hook question from the very beginning. If adenine is 30%, then thymine has to be 30% too. That's 60% of the total, right? So the remaining 40% has to be split equally between guanine and cytosine, which means you get 20% for guanine and our answer 20% for cytosine. Now watch out for this trap. Always make sure your final percentages add up to 100. Now, let's clear up another common point of confusion, the 2 types of bonds holding DNA together. First, you've got the weaker hydrogen bonds. These are the ones that form between the base pairs, kind of like the glue holding the 2 strands together. Then you have the super strong phosphodiester bonds. These form the backbone of each strand, linking one sugar to the next. The key takeaway, hydrogen bonds are between strands. Phosphodiester bonds are within a strand. Don't you dare mix those 2 up. Okay, so we finally understand the thread. Now, how do we pack it? Well, in simpler organisms like prokaryotes, which don't have a real nucleus, the circular DNA is just kind of supercoiled in a region called the nucleoid. But in eukaryotes, like us, the system is way more elaborate and, honestly, a lot more elegant. The entire solution lies with these amazing proteins called histones. I want you to think of them as tiny, positively charged spools. Now, since DNA has a negatively charged phosphate backbone, it's just naturally attracted to these positive histones. It's a perfect electrostatic attraction that lets the DNA thread wind super tightly around them. Simple physics. The core of this spool is made of 8 of these histone proteins. You get 2 each of H2A, H2B, H3, and H4. Together, they form what's called the histone octamer. Then another histone, H1, comes along and acts like a clip, locking the DNA onto the core. This whole complex, the DNA wrapped around the histone octamer, is called a nucleosome. And how much DNA actually wraps around each core? Well, it's a very specific number, and you should probably remember it for your exam. 146 base pairs of DNA make about 1.65 turns around each of those histone octamers. This slide shows the entire process just beautifully. You start with a raw DNA thread, the double helix. That thread then wraps around the histone beads, creating this famous beads on a string structure. But it doesn't stop there, oh no. These nucleosomes then coil up even more tightly into a fiber, which then loops and folds with the help of even more proteins, until, finally, you get the highly condensed chromosome we see during cell division. It's just incredible. So why does all of this matter? I mean, why go through all this trouble? This intricate packaging is about way more than just cramming DNA into a tiny space. It's really about control. But before we get into the why, let's lock in the what with a couple of quick memory tricks. For the base pairing, just remember the letters A, T, G, C. For the base families, think pure as gold. The purines are adenine and guanine. And for that histone core, just remember the 4 numbers, 2A, 2B, 3, and 4. Got it? Awesome. Now let's talk about the real power of this packaging. Okay, the power. First, it obviously solves that massive storage problem we talked about, but crucially, it also allows for gene regulation. You see, tightly packed DNA is basically turned off, while loosely packed DNA can be read by the cell and turned on. This is why errors in packaging are directly linked to some really serious diseases. Now that you've got the theory, let's practice. Pause after each question and try to solve it yourself. Question 1. Who proposed the double helix model? Is it A, Griffith, B, Watson and Crick, C, Darwin, or D, Mendel? The answer is B, Watson and Crick. Question 2. In DNA, adenine pairs with A, guanine, B, cytosine, C, thymine, or D, uracil. The correct answer is C, thymine. A common mistake here is choosing uracil. Remember, uracil is an RNA, not DNA. Question 3. Let's do a match the following. List I has H1 and H3. List 2 has core histone and linker histone. The correct match is H1 is the linker histone and H3 is part of the core. Question 4. Which of these statements is correct? 1. DNA strands are parallel. 2. Histones help in DNA packaging. The only correct statement is number 2. And this catches a huge mistake. So many students forget that DNA strands are anti-parallel, not parallel. Last one, an assertion, reason. Assertion, the percentage of adenine equals thymine. Reason, A pairs with T through 2 hydrogen bonds. The answer here is A. Both are correct, and the reason is the perfect explanation for the assertion. All right, let's do a super quick fire revision. Ready? Double helix model, Watson and Crick. The golden rule, A equals T, G equals C. Purines, that's A and G. The phosphodiester bond builds the backbone. The histone octamer has H2A, H2B, H3, and H4, with 146 base pairs wrapped around it. Phew. Now, how did you do on those practice questions? Go ahead and comment your score out of 5 below. If you scored 4 or 5, you are totally ready for this topic. If you scored less than 3, just give this explainer one more quick watch. And this final question on the screen, well, that's the key to understanding the next level of genetics, gene expression. You did great work today, and I'll see you in the next one.